Hello guys, so let's start with a new topic, central dogma. Now central dogma, it is a process starting from the DNA replication till protein synthesis. So the first step in this is DNA replication. So write down. I would suggest you to take 2 pages for writing this because we will be writing it very long like this, okay? So I suggest you to take a middle page or somewhat 2 pages side by side so that it will be easy for you to write, okay? So write down DNA replication. The first step is DNA replication. Now, DNA replication means what? Duplication of the DNA, making copies of the DNA. Okay. So, the type of DNA replication which happens in human is semi-conservative type. It's called a semi-conservative type, and the reason for that is, let's know right now. So, suppose this is a DNA strand. Okay. Suppose this is a DNA coiled upon 2 strands coiled on each other. Now, there will come an enzyme, DNA helicase, and it will separate these 2 strands from each other. Okay, so it will separate the 2 strands from each other. So this becomes one parent strand and this becomes another parent strand. Parent strand, we take it in terms of maternal strand. Okay, so we call it as one maternal strand and this is another maternal strand. Then will come one more enzyme, that is DNA polymerase. It will start to form another DNA. Okay, the copy of this strand, this maternal strand copy will be formed, and the copy which is forming is called as daughter strand. So this copy which you're looking at, this is daughter strand. This will happen on both the maternal strand. Okay, so this will happen on both the maternal strand. Now you can see I have not made it in one line, I have made it into fragments because the daughter strand which is formed, it is formed into fragments, and those fragments are called as Okazaki fragments. This is a short overview of semi-conservative type. There's a lot of thing inside it, 5' end, 3' end, and a lot more thing. For Krok, you should only know the process, the overview of the process, okay? So, daughter strand is formed in Okazaki fragments. Now, this Okazaki fragments or these DNA fragments, they will be joined with another enzyme. The enzyme name is DNA ligase. We'll be doing all these DNA enzymes in the next video. For now, just remember this process, okay? So, they will be joined with Okazaki fragments, okay? So, DNA ligase came and now the strand is joined and formed into one strand, okay? And it will also similarly coil upon DNA. So, suppose, so you can see a new DNA is formed. It is this DNA, once the Okazaki fragment is joined with DNA ligase, these strands coil over each other and this becomes your new duplicated DNA. Similarly, this will happen with the second maternal strand which was present. So you see from one DNA, we made 2 DNAs. So from one, we made 2 DNA. This is called as DNA duplication, making copy of the DNA. Okay. So I hope this is clear, this DNA. And the reason why it is called as semi-conservative, now let's write down the semi-conservative because half of the information comes from or comes through maternal chain. Right, I said you the maternal chain will make a copy of it, right? Here you can see this maternal chain is making its copy, right? So the information is conserved, but half conserved, right? These 2 strands because got divided into different strands, so half of the information is conserved and a new strand is, daughter strand is formed, and similarly a new DNA is formed. So half of the information comes through maternal chain, and that is the reason we call it as semi-conservative type. I hope this is clear. Meaning of semi-conservative is that half of the information is through maternal chain. Now let's do the second step in central dogma. Once DNA is replicated, we will form mRNA from the DNA. Okay, so from DNA we are making mRNA, and that process is called as transcription. And what is transcription? We are forming mRNA from what? We are forming mRNA from DNA. So for transcription, you can write down DNA to mRNA, meaning of it, okay? And we require an enzyme for this process. The enzyme name is DNA dependent RNA polymerase. It is also known as only RNA polymerase. We call it as DNA dependent because depending on the DNA, the mRNA is formed. And we only call it as RNA polymerase sometimes because we are forming the RNA polymer, okay? So mRNA is formed. But after transcription, the mRNA which is formed is actually an immature mRNA. So write down this mRNA. It is immature mRNA. You can also write the 3 steps of transcription. So write them down. The 3 steps of transcription are initiation, elongation, and termination. Okay, so these are the 3 steps of transcription. Then what is formed? An immature mRNA is formed. For making this immature mRNA mature, we do a process called as processing. So we do processing. First write down this immature mRNA. Its another name is hnRNA. hnMRNA. HN is heterogeneous mRNA. Okay. Now the HN or immature mRNA contains what? It contains exons, introns, exons, introns. Similarly, this process goes on in the whole mRNA fragment, the immature mRNA strand. Okay. Such an mRNA which contains this structure, exon, intron, exon, intron, exon, intron, and so on, is called as immature mRNA. So we do processing to make immature mRNA mature mRNA. So write down the meaning of processing. Immature mRNA to mature mRNA. So processing, it has 3 steps. Now you can write down these 3 steps like this: splicing, capping, and tailing. So splicing is removal of introns. Removal of introns means exon, 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 so on. This will be the structure of the mRNA once the introns are removed. Exon, exon, exon. Second, for capping, at the 5' end of DNA, we add methyl group. DNA has a 5' end and a 3' end. Suppose this is a DNA, so suppose this is a 5' end, this is the 3' end. That's all you have to remember. So at the 5' end, we will add a methyl group. And tailing means at 3' end, we add polyadenyl group. Polyadenyl group. And once these 3 steps are done, the splicing, capping, tailing, we get a mature mRNA. What do we get in return? We get a mature mRNA. After these 3 steps, splicing, capping, and tailing. Sometimes they ask you in the question the structure of the mature mRNA. They'll give you many options, and this, the correct one will be exon, exon, exon. If they ask you immature mRNA structure, then it is exon, intron, exon, intron. Let's move to the next step of the central dogma. Once the mature mRNA is formed, we go for protein synthesis. Okay, so we go for protein synthesis, and that process of forming proteins is called as translation. Okay, so and for translation, we require 2 things, very important: magnesium and ribosomes. In the organelle video, I told you that ribosomes is the place where protein synthesis occurs. So these are the 2 things. Magnesium, it acts as a cofactor. Magnesium is a cofactor, and ribosomes are required for protein synthesis. So translation is synthesis of protein. Translation means what? Synthesis of proteins. So what are formed at the end? Proteins are formed. Okay. Various amino acids are formed, and these amino acids together comes and form a polypeptide chain, which is protein. Okay. So write down many amino acids forms a polypeptide chain. That is proteins. Sometimes they give you the question that where does protein synthesis occurs? So the answer is polyribosomes. The polyribosomes or polysomes means ribosomes. You can write this point, many ribosomes together. You can write this point extra somewhere on the page, wherever you have space. Many ribosomes together is called as polyribosomes. Poly means many, or sometimes, rarely, they give you polysomes. Okay. So remember this point also. I hope this central dogma is clear, which consists of DNA replication, which is of semi-conservative type in human. Then once the DNA is replicated, we move to formation of mRNA from DNA, and that process is transcription. Now we know all the 3 steps of transcription. For translation also, we have these same 3 steps: the initiation, elongation, and termination. Okay. Once transcription is done, we get an immature mRNA. to make it a mature mRNA, we do processing. This processing, it consists of 3 steps: splicing, capping, and tailing. Once these 3 steps are done, we get a mature mRNA. Mature mRNA then goes for translation in the ribosomes. So in the ribosomes, magnesium factor is required. So along with magnesium, the proteins are formed. Proteins are nothing but many amino acids are formed over there, which together comes and make a polypeptide chain, that is your protein. Okay. This processing, you can add a point over here, that this processing, it is also known as post-transcriptional process. It happens after transcription, right? So post-transcriptional process. Okay. So I hope this central dogma is clear. Now write an extra point regarding this RNA polymerase. So let's write it over here. RNA polymerase are of 3 types. It's very important for Krok point of view. So they are of 3 types. So write the type 1, type 2, type 3. RNA polymerase forms RNA polymer. Okay. So type 1, it forms rRNA, that is ribosomal RNA. Small r stands for ribosomal. Type 2 forms mRNA, which we studied just now in the central dogma. Here M stands for messenger RNA. And the third type is tRNA. Here T stands for transfer RNA. Okay. Among this, if someone eats a poisonous mushroom, the name of it is amantadine. If someone eats this poisonous mushroom, in these people, transcription is blocked. Now, how is transcription blocked? This amantadine, it blocks RNA polymerase type 2. So, type 2 is responsible for forming mRNA. If mRNA is not formed, so the process of forming the mRNA is transcription. Therefore, transcription is blocked. So, write down amantadine. It blocks type 2 RNA polymerase. Therefore, transcription is blocked. The DNA replication enzyme we will be doing in the next video. So that's it. Thank you.